Welcome, everybody. My name is Devin Lau. I'm the Associate Director for Yale Center Beijing, and we're very happy to have you join us. Um, the center was established in 2014 as a way for Yale to increase its engagement with China, and we are the first and only Yale University-wide center. So we service the whole university, and we like to feature people from all sorts of academic fields to have discussions here. And we've been lucky to have people from the med school, as well as from everything from drama to art history to classical music to economics and politics. So a very eclectic mix of people. And we're very excited to have a talk today that'll apply to everybody because it's about aging and health, and we're all aging. So and we all want to be healthy. So this is going to be a good opportunity for us to all hear from Professor Vishwa Deep Dixit, who is the Waldemar von Zedtwitz Professor of Pathology, Comparative Medicine, and Immunobiology, as well as the Director of the Yale Center for Research on Aging. He's studied veterinary medicine in India before completing his PhD at the University of Hanover in Germany, as well as doing postdoc research at Morehouse School of Medicine and the National Institute on Aging at the NIH. His lab studies immunometabolism and aging, focusing on understanding the interactions between metabolic and immune systems with the goal of revealing molecular targets that can be harnessed to control inflammation and immune dysfunction as a means to enhance health span. And so hopefully he'll explain a little bit more about what exactly he's doing. And I'm sure many of us are eager to hear how we can increase our immune systems and metabolic systems to help us increase our health span. So I'll turn things over to Professor Dixit.
All right. Thank you, Devin. Thanks for your kind introduction. Let me try to share the screen. Okay, yeah, great. Thank you, Devin, and thanks to all of you, all of my colleagues. I believe most of you are currently in China, so good morning from New Haven. I believe it's evening for most of you, so it's great that we can all be connected, even though perhaps even though it's not in person. At least we are connected virtually. So thanks for joining. And like Devin mentioned, my laboratory studies aging, and I'm going to kind of give you a broad overview of what we are doing and share some data that we have recently published. Okay, so I guess Devin mentioned that we have a quite a broad group of people, very group of audience with diverse backgrounds. So I think I'm going to try to keep the presentation as simple as possible. And hopefully at the end, you can have questions and I'll be able to answer and provide any clarifications. Okay, but just want to remind everyone, first and foremost, is things that often get lost when people talk about risk, okay? Risk and disease. So people will say, okay, if you drink too much, if you smoke too much, if you're going to be overweight, you have much more chances of getting cancer, right? Indeed, that is the case. But if you compare the risk, the relative risk of getting cancer, all type of cancers, and compare these risk factors with aging, the process of aging, you find that aging by itself is strongly associated with increased risk of disease, okay? And this risk of disease that is caused by process of aging is across the board, actually, okay? So for example, if you look at heart disease, you find almost the same thing. I'm sure your doctor has told you that, if you have hypertension, if you're overweight, again, smoking, you're going to have high chances of getting heart disease. But if you add aging, or if you just have aging as an independent risk factor, again, you get the same result as cancer. And obviously, a major disease that is induced by aging or caused by aging or is a risk factor, where aging is a risk factor, is Alzheimer's disease. On the left is a graph which shows what is the relative risk of various diseases as we get older. Okay. And I think it's very easy for all of you to see what is happening here, right? Something happens as we hit somewhere around 45, 55, right? That the incidence of chronic diseases in humans increases quite dramatically from that point onwards. Okay. So these are all the diseases that are listed: heart disease, cancer, stroke, emphysema, pneumonia, diabetes. We also added accidents because people fall, obviously chronic kidney diseases, and Alzheimer's. So what is it? Okay, what is it that is caused by aging? Or what does aging do, the process of aging do, that increases the risk of all these diseases simultaneously? Okay, something must be happening. And the idea in the field is, for example, let's say if we develop a drug just specifically for Alzheimer's disease, okay, and we magically cure Alzheimer's, process of aging will still continue, and you would still get other type of diseases if you live long enough. So in other words, people think that and that's actually what's happening in the biomedical research enterprise all over the world, is that we treat each disease at a time. I know this is important, and that's one of the ways to kind of tackle these conditions. But another idea has been that instead of treating each disease at a time, which are all age-linked, that we would eventually get, if we treat diabetes, we'll get, cancer, or if you treat cancer, you'll get heart disease. What if we can understand what is happening in this process of aging, so that maybe we can even target those mechanisms and delay the process of aging, so that one can prevent onset of all the diseases at the same time, if we do find that critical mechanism of aging. So that is, the big idea in the field world over, that people are quite interested in, and my laboratory is trying to understand what is the mechanism of the process of aging that impacts various organs. And our primary interest currently is in immune and metabolic systems, for the reasons shown in the next slide. And the slide, okay, is here. So here, what you're looking at on the upper panel are what we call the hallmarks of aging. Okay, the key mechanisms that we think are responsible for causing process of aging itself. Okay, among these hallmarks that are listed here, inflammation is one of the major hallmarks that we are quite interested in, a process whereby there is increase in production of pro-inflammatory cytokines that happens with age in humans and in many animals. There is increase in molecular damage, and there's the damage products accumulate in the cells, which also cause inflammation. This is called as damage-associated molecular patterns. Then you have cellular stress, mitochondrial stress that produces reactive oxygen species. There is metabolic dysfunction that happens with age, and visceral adiposity is, one of the major hallmarks and risk of these factors. There is impaired proteostasis that is one of the mechanisms of aging, where there is reduction in our ability to clear damaged proteins, a process called autophagy. There's also stem cell exhaustion, that our overall stem cells, either they're exhausted or they're lost, or the niches where they're present are dysfunctional, so that we are not able to regenerate things that we can do very easily when we are younger. Okay. And then there is genetic changes, including methylation changes in the genome, that alter the transcription of the genes that control various biological process, and then these global changes somehow cause this process of aging and leads to this loss of resilience. Okay, but then you will ask me, or my grandfather is 94 years old. He doesn't have any of these diseases, but my neighbor is 60 years old and he has, let's say, gout or heart disease. So we all know that aging, nobody gets all the diseases at the same time. So what is actually happening? So we think that when these mechanisms are in play, what the host does is it kind of loses its homeostasis, or the process where all the systems are functioning at its best level. And so there is a loss of resilience. Resilience means ability to rebound. Okay. But then just the loss of resilience is not going to cause a disease. Okay. So for that disease to manifest, you have to have specific associative triggers. Okay. For example, I'll give you a very clean example, which is disease called as gout. Okay. Gout is a disease that is age-related disease, but we don't get gout. We, of course, and young people don't get gout, old people get gout, but gout would only happen if you have uret crystals that are present in the joints. Okay. Same thing is with cancer. For cancer to manifest, you have to have these perturbations in oncogenes, xenobiotics in the background of aging. You have to have associative trigger like dyslipidemia for heart disease to manifest. For Alzheimer's disease, although there is still debate, presence of amyloid beta is considered really important for disease to happen. And similarly, you will only get infection and disease with aging if you have a pathogen that is present. You have to have a virus or you have to have a bacteria to be present to cause that disease. So, so what we are trying to understand is if we can understand this process upstream here, is it our, and if we do that and we improve the ability of the host to maintain homeostasis and resilience, that in that case we may be able to actually prevent multiple age-related diseases that occur. Okay, and if you really think about it, this is really not a very new idea. Okay, this is a picture of Ilya Mechnikov, who discovered macrophages or phagocytes and really laid the foundation of innate immune system, innate immunobiology. He wrote this book called as Prolongation of Life Optimistic Studies while he was in Pasteur Institute in Paris. If you get a chance, you should read this book. Really very interesting book. So, by the way, Ilya Mechnikov was also the person who coined the term gerontology, okay, the study of aging. And Ilya Mechnikov predicted several years ago that, and this is the excerpt from his book, that he predicted that the phagocytes or macrophages are going to be really important for the diseases of aging. And he said that the phagocytes resist the influence of invading poisons better than any other cells of the body and sometimes are stimulated by them. Okay. And you would be wondering, oh, what are these poisons and what does he really mean? And remember, this is what he said that almost 120 years ago. So at that time, we had very little knowledge of modern biology and modern molecular biology. Of course, immunology was basically non-existent. So what Ilya meant with poisons is shown in this electron micrograph image of a phagocyte or a macrophage that is isolated from an old animal. Okay. So what you see here is this phagocyte that is actually accumulating these poisons. And you will say, which are the poisons? And the poisons are pretty obvious here. So this is a macrophage that has phagocytosed or potentially accumulated lipid in this big lipid droplet. I mentioned that there is impaired autophagy. So this is increase in protein deposits, protein aggregates within the macrophage. This is the nucleus of the macrophage. In this case, it is misshapen. And then you can see these so-called poison-like things inside these lysosomes, which are electron-dense material. And then you also find these weird structures in the cell. And this is a crystal, okay? These are the kind of poisons that Ilya Mechnikov was talking about. And now we have modern words for that. We call them damage-associated molecular patterns, okay? And these damage-associated molecular patterns, once they accumulate in cells like macrophages, macrophages can actually sense them, okay? And when macrophages sense them, then macrophages produce inflammation. And without going into too much details, I will just summarize years of work that what happens with age is there is a very special sensor, multi-molecular platform in the macrophages called as the NLRP3 inflammasome, which was first discovered by Jörg Chopp in Switzerland. This molecular pattern recognition receptor can sense many type of different poisons or different damage associated molecular patterns that are listed here. And that's the interesting part about this molecule, because with aging, what we get is increase of multiple type of damage associated molecular patterns or these poisons, okay? And inflammasome can get activated by many of these things. Now you would ask, oh, well, if that is the case, why would, biology have, or why would we even have this molecule? Obviously, we don't want to get disease all the time. No, actually, this molecule is present to protect us from infections. However, in the process of aging, what we get is this continuous slow cellular damage, and macrophages try to clear it by activating this complex. But the damage kind of continues because process of aging, time continues, and what it does is basically continuously activates this complex. And when this complex is continually activated, what it does is it cleaves this important enzyme called as caspase-1. And caspase-1 would then activate two very critical cytokines, IL-18 and IL-1 beta. And these two cytokines then produce inflammation. And when we block these NLRP3 inflammasome genetically, then you can actually prevent the process of inflammation, at least in mice. And once we do that, what you also get is inhibition or protection of animals from many diseases. So the animals, the mice that are old and have reduced activity of NLRP3 inflammasome are protected from bone loss. They are also protected from diabetes, and they have improved immune system because they have better thymic function. They also have greater memory in old age because they have less cognitive decline and less inflammation in the brain, and they are less frail. And then you would wonder, wow, this is like sounds pretty interesting. How come there are effects on so many different things? And I think the answer is truly what Mechnikov had predicted, is it's the phagocytes. So macrophages are present in all these different locations, as tissue resident cells. And I think what is happening is with aging is that these cells are getting activated through the process of this this molecule called as NLRP3 inflammasome. Okay. So one of the things we are interested in is to find where does inflammation originate. Okay. What is the source of inflammation? Organ source and also the cellular source. Okay. So what you're looking here is a CT scan of a healthy person and 31-year-old, and actually also a healthy person that is 74-year-old. Okay, there is no, and both these people have the same amount of weight. Okay, but there is one big difference. I think you can see it very clearly in this image, right? You see the difference? Yep. So the older people, as we get older, we accumulate fat, okay? We accumulate fat inside our viscera or where our organs are. It's called visceral adiposity. You don't have to be actually obese to accumulate it. obviously, once there is obesity and overweight, you would accumulate fat inside your stomach. However, even if you maintain a normal body weight, you would still accumulate fat inside your organs called visceral adiposity. Okay. And it is known now that increase of fat both in men and in women with age, as shown here, is a major risk factor for several diseases that emerge with age. And what we're trying to understand is it possible that increase of this visceral fat is causing inflammation with age? And is that the part of the mechanism of process of aging itself? And okay, so this is a slide which is very interesting in my view, because what you're looking at here is everything that is green is a myeloid cell, which is mostly in this case are macrophages and a few neutrophils. And if you would show this figure to any pathologist and ask, what is this? They will say, oh, this is an immune organ, because everything that's green is a myeloid cell, and myeloid cells are immune cells, and there's so many green cells, so it would be an immune organ. But the answer is it's not an immune organ. It's actually adipose tissue, your fat tissue. And what is quite interesting is that the fat tissue in normal situation, in healthy situation, actually harbors lots of these macrophages that are present in the fat. And what these macrophages do is they maintain the normal function of the adipose tissue. And we're trying to understand with age, as aging happens, what happens to this organ, which increases with age? Are there changes in the immune system? And are these immune cells aberrantly activated to cause increased inflammation? So here is some data to prove to you that indeed adipose tissue is an immunological organ. So you're looking at single cell sequencing analysis here of young visceral fat from a healthy mouse and an old visceral fat from an aged animal. Okay, and when you do the single cell analysis and look at these UMAPs, this work was done by my former colleague, Emily Goldberg, who is now faculty in University of California, San Francisco. So what Emily found was that in young animals, there are lots of different type of immune cells that are present within the adipose tissue. Okay, so this is, for example, cluster of a very specific, very interesting cell called as innate lymphocyte cell 2. There are macrophages present. There are different type of macrophages. There are NKT cells. There are gamma delta T cells. There are T cells. There are B cells. Every type of immune cell that you know of is present, except neutrophils and eosinophils. Okay, but what happens with age is very dramatic. So some cells are actually completely lost. So for example, if you look at the cells called as ILC2s, they actually disappear. Several other cells also we lose with age, for example, NKT cells. And macrophages that I've been telling you about, in fact, also go down. Okay, remember Mechnikov had told us 100 years ago that phagocytes help clear the poisons from the body, okay, help clear the debris. And it seems that, we are losing those cells that are helpful to us with age. And this leads us to definition of what are the cellular hallmarks of immunological aging. What happens to the immune system with aging? Okay, and few of those are listed here. One is that we and others have shown that effector T cells expand with age, including the suppressor cells or the T regulatory cells. They also increase. There is expansion of a specific type of B cells called as a pro-inflammatory aged B cells, and this happens in unique structures called as fat-associated lymphoid clusters. This happens in the adipose tissue. There is a loss of natural killer cells and the natural killer T cells, and there is a loss of innate lymphocyte cells too, ILC2s, and there is also a reduction of a specific type of dendritic cell subset and the homeostatic macrophages or phagocytes. Okay. So the question is, when all these things are happening, how can we actually study if we can block those things, if we can block the inflammation? And And the one of the things that I have been interested in for several years in my lab is this process where there is negative energy balance in a host. In other words, when you eat less, okay, when energy consumed, which is this is basically your food, okay, you get energy by consuming food. If you consume less food and you expend more energy, so in other words, if you're eating less, walking more, running more, this would cause your energy balance equation to be in negative, which means that you need more energy, but you're consuming less of it. Okay, when this happens, okay, what you actually get is increased lifespan. Okay, this has been shown in many species, and through a process called as caloric restriction. If you restrict calories, mice, rats live longer, flies, worms live longer. There is some indication that even monkeys may live longer. Okay, but one of the things that you have to imagine, when a host is in negative energy balance, it has to do many things, right? It has now be able to judiciously use the remainder of the fuel it has. Fuel, these are all energetic substrates. So immune system and metabolic system have to have this coordinated dance so that it can maintain homeostasis. There is one problem, however. Okay, when you get increased lifespan by restricting calories, the problem is of trade-offs. Okay, so there are trade-offs with this increased lifespan, so that you have reduction in growth, your reproductive system is less active, immune system is less active. So basically, in other words, energy is diverted from less essential functions to more essential functions, which is like running the heart, running the brain. And people think that the longevity that you get in because of caloric restriction is because of these cellular trade-offs, okay, also organismal trade-offs. Now you would ask, okay, if we are going to have a trade-off, if we're going to live longer, but if we cannot grow and if reproduction is bad, we are getting infections, what's the point of living? I fully agree with you. So the question is, how do we study this mechanism in a way where there is no trade-off, but we can understand what is driving these positive effects? So, and then, it is known, like I told you, that calorie restriction increases lifespan. But then there was a paper that showed that several mouse strains actually not only live not longer, they actually live shorter. Okay, so some mouse strains live longer, some live shorter. There was a big debate, and everybody got really confused whether calorie restriction is really real or whether it is just some sort of an artifact of a laboratory condition. Because we know that the animals, when they are housed in animal houses, they're in a very different condition as compared to what we live, because the animals live in pathogen-free environment. They don't see any pathogens. And if you take a mouse or a worm or a fly and stop giving it, it's 40% of the food, animal does not know what is happening, because animal does not sign consent forms. So animal undergoes the stress response. So a lot of these things then cause changes in the physiology, so that it is very difficult to understand what is actually happening during calorie restriction. So the idea was to actually do studies in human beings, okay? Do a calorie restriction study in humans. And then if we find that calorie restriction in humans is biologically relevant, then we identify endogenous targets that are affected by CR, and then use those targets to assign causality, to see whether they are just, really, if they're passengers or if they're drivers. If they're important, then we can test causality using model organisms and see whether they're important for mechanisms of disease or lifespan itself. And this is what we did. So this was a study called as Calorie. It was a clinical trial that was done at 3 different sites. At Baton Rouge, this is my former institution. I used to work here before I came to Yale. In St. Louis and Washington University, in Boston, in Tufts University. And what the study wanted to do was basically take people that are healthy between 25 and 45 years of age. They're not old people, and restrict the calorie intake of the individuals by 25%. So you eat 25% less food for 2 years. Okay. And then after 2 years or during that time, study whether calorie restriction or negative energy balance is reducing the risk of disease. And here is the data. Remember, the clinical trial wanted to restrict people by about 25%. But in fact, when you take people and you tell them to restrict calories and tell them to go about their life as normal in free living condition, human beings, it seems, can only restrict about 15% calories, okay, not 25%. And this is the percent of calorie restriction in people. This is baseline, which means at this time of the study. M12 means 12 months of after the study, and 24 months, so year 1 and year 2. And what this study initially showed were there were some markers of aging and oxidative damage which were reduced when people underwent calorie restriction. So which was quite exciting, suggesting that there may be indeed be that calorie restriction may be beneficial to people. And then the question was, to what should we study that actually tells us that there is effect of calorie restriction? Because remember, I told you that these are all healthy people. Okay. I also told you they are not old people. So if somebody is old and is young and healthy, what are you going to study that is going to tell you that something is working? Okay. Luckily for us, for immunologists, there is an organ that ages faster than any other organ of the body, and this organ is called as the thymus. And the thymus is a key organ, as you know, that produces T cells. And what you're looking at, the magnetic resonance imaging of a healthy 25-year-old person and a 45-year-old person on the right. And if you do the MRI, this is the heart, it's the aorta, this is the sternum, and this area here is the thymus. Okay, and this white spot where my arrow is the fat inside the thymus. Okay, let me find it for you again. This right here. However, if you look at somebody who is just 45 years of age and healthy, you can see that all the thymus is white. So this is the heart, is the sternum. So the 45-year-old person, about 80% of the thymus is all made up of fat. Yes, in healthy people. And it's highly conserved throughout evolution in vertebrates. This is the same image of this person. Here we have basically blocked the signal that is coming from fat. So the only signal that is coming from is water. So with that, you can actually calculate the functional volume of the thymus. So this is the thymus in a 25-year-old person. You can clearly see it is present. And in a 45-year-old person, there is nothing or almost nothing. So we wanted to study what does calorie restriction do to the thymus. And the effects were really very striking. And this work was done by my colleague, Dr. Yuni Yum, who's an associate research scientist in the lab. And what she found was that when you study people at the baseline and then after 2 years of calorie restriction, you see that overall people have lost some weight. This is white, is all weight, basically fat. But if you look at the thymus, this is the same person that came at baseline and after 2 years. And when you look at this, actually the size of the functional thymus is actually bigger. Okay, so somehow calorie restriction in human beings is in fact rejuvenating the function of the thymus, increasing the function of the thymus, not just protecting, but actually rejuvenating, reversing, which was quite surprising because we did not really expect this to happen. So this is just the quantification of the thymic volume, the functional thymic volume. There is a marker that can tell us about new T cells that are made by thymus. Just because the size of the thymus is bigger doesn't mean the thymus is better. So you have to measure whether the thymus is making new T cells. So there's an episomal DNA which we can measure in T cells that indicates presence of new T cells. So you can see that when people come at baseline, but after 2 years of CR, they all have increase in this marker. So in the next few minutes, I will quickly, show you the data and not go over all the details, but give you some big picture of what we're trying to do. So obviously, we're trying to understand how does calorie restriction works, what is the mechanism, in this case, for reducing inflammation. And then if we find that mechanism, can we actually use those targets to then develop maybe new drugs against inflammation and potentially against aging? So we collected the adipose tissue from people at baseline after 1 year of calorie restriction and after 2 years of calorie restriction, and did the transcriptomics analysis. And what is really striking is the differences that exist in people after calorie restriction. So these are the genes that are inhibited. So you can see that people have a dramatically homogeneous response in this case to this intervention. Several genes that are up and down. And these are the pathways that are really interesting that are changed in human beings. So these are pathways that are increased by calorie restriction in human beings. We think that these pathways may even be important for lifespan itself, maybe increasing the lifespan, because some of them have previously been shown in worms to be associated with increased lifespan. And then there are pathways that are reduced, okay? And the pathways that are reduced are mostly in the pathways that are in inflammation, okay? And one of the genes that we discovered to be really interesting in this case is this protein, this gene called as PLA2G7, which is inhibited by calorie restriction. This is a very interesting molecule which controls the lipoprotein homeostasis. It is mostly produced by the macrophages and controls hydrolysis of platelet activating factor. Okay. So this protein is, or this gene and protein is present very high in very high amounts also in the plaques in the heart disease. And there have been some studies previously showing that if you reduce PLA2G7, it can reduce some inflammation. So we were very interested to see whether PLA2G7 is one of the molecules that is inhibited by CR, but maybe a target for potentially reducing inflammation. So what we did was we created the mice that lack PLA2G7. So these mice are more or less normal here, wild type and knockouts. But if you give the wild type animals in black the diet, a high fat diet, or what we call the Western diet, you see that there is an increase in body weight. There is mice become fat. But the red are the knockout mice that have reduced amount of PLA2G7, and that these animals are partially protected from obesity. I will not go into the details, but just to quickly tell you that PLA2G7, if you reduce this amount, you reduce the comparing this band to here, this is active caspase 1. So reduction in PLA2G7 reduces the NLRP3 inflammasome. Remember that the molecule, the pattern recognition receptor that I told you right from the start is very critical for aging and inflammation. And here is the structure of the thymus. So remember, thymus ages faster than all other organs. It becomes smaller, and the thymus basically loses its architecture. There is a lot of fat that's present. Cortex and medulla are obliterated. And if you compare wild type animals on top to the knockout animals on the bottom, you see that the thymi are better. You can see the structure of these animals is much better. And so this work was published early this year, and this is a commentary that was written by my colleagues, basically suggesting that if you reduced PLA2G7, what we found that in this case, you can mimic several aspects of calorie restriction in mice. So remember, this is a target that we found from humans first, okay? It is associated with improvements, but then we reduced it to show causal effect, that it actually improves function in many of these disease conditions, okay? So here, the key point is for you to remember is that the immunometabolic response that is induced by calorie restriction in humans can produce this anti-inflammatory target called as PLA2G7, okay? And this study has also shown that calorie restriction in human beings actually works, okay? And it can, in fact, rejuvenate certain organs that we thought are lost forever. For example, the thymus, by increasing thymopoiesis. I will end because we are running out of time and I don't want to overload you with too much data, but just summarize one more target that we discovered. I will just show you the summary slide of that, which is this target called as protein called as Spark. Okay. This protein is also inhibited by calorie restriction. This work was done by my colleague Sunjin Roo, who just started his own laboratory in Korea. So what Sunjin found was that when people are calorie restricted, the levels of spark go down. Okay, but here the spark is made almost exclusively in humans, or not exclusively, but I would say predominantly by fat cells or adipocytes. So when one is in negative energy balance or calorie restriction, the levels of spark go down. When you are in positive energy balance, like in obesity, when there is too much calories that you're consuming, spark levels go up. Okay. When spark levels go down, it reduces inflammation, it increases the tissue repair, and also promotes homeostasis by increasing the anti-inflammatory response in macrophages. Okay. When spark levels are higher, then spark activates these macrophages, making them more pro-inflammatory, producing more inflammation, reducing tissue repair and impairing the tissue function. How does spark work? So what Sunjin Roo discovered was that adipose tissue makes spark, and this spark acts on toll-like receptor 4. And when it does that, it activates two arms of the TLR4 pathway. Okay, one arm is noted here, TREF-TBK1 IRF pathway, and what it does is actually induces interferon response, okay, which is a very interesting molecule involved in controlling viral infections, but also important in causing disease. And when it activates the MID88 arm of the TLR pathway, Spark then activates pro-inflammatory cytokines, IL-6, IL-1 beta, and TNF-alpha, causing more inflammation. So what we found was that if you can reduce Spark, which decreased in people upon calorie restriction, then you can actually reduce inflammation. And here is the mechanism that I just explained to you. So the take-home point of this slide is here that first you have to, if somebody argues whether calorie restriction is relevant to human biology or not, I think the answer is here, and the debate is over. Indeed, CR or negative energy balance is very relevant to human biology, as has been shown from this clinical trial. Other thing very interesting is that you just have to limit your calorie intake by just 15%. Okay, it doesn't matter what you're eating in this case or when you're eating it, which obviously not everyone can do. And what we are trying to do is study how this happens so that we can identify how the mechanism of CR can it be harnessed so that we can identify new targets or new drugs that could give us the beneficial effect of calorie restriction without actually having to do calorie restriction. So I showed you the data from two of these molecules, PLA2G7 and Spark. And I think only time now will tell what will happen when we do clinical trials with these two molecules, whether it is clinically beneficial or not in human beings or in aging individuals. So I will stop here. And I have thanked people that have done this work as I went along. Most of this work obviously cannot be done unless or until I have this amazing group of people, my colleagues, students, and postdocs. The most of the calorie restriction work was done in the lab by Yoon-hee Yoon. The identification of the changes in the adipose immune system was done by Dr. Emily Goldberg. The work in relation to identification of spark was done by Dr. Olga Spadaro, and who is now working in AstraZeneca as a senior scientist. And the identification of spark as a CR mimetic. This work was done by my colleague Hyunjin Roo, who is now running his own laboratory, and several of my colleagues at Yale and in several other places in the country. So I thank you very much for your attention. I will be happy to answer any questions that you may have, and I will stop sharing the screen so that you can see me, and if you have any questions, you can ask.
Great. Thank you so much. Such amazing research that you're doing. And I'm sure there are lots of questions that people have. So feel free to raise your hand and I can call on people, or you can leave your questions in the chat box, and we'll try our best to get to all of them. And I see that Yang Yue has their hand up, so let's start with Yang Yue.
Thank you so much for your sharing your expertise and knowledge. And I have 3 questions. First one regarding the aging. So what's your opinion about the inflammation and aging? Which one is cause and which one is effect? I feel like it's sort of like the chicken and egg. And the second question is, in your study, you showed the adipose tissue. I wonder which one, like white adipose tissue or brown adipose tissue you looked at. And my third question is that calorie restriction shows really promising effect. I wonder whether you also looked at the individual metabolism, because given that everyone has a different metabolism and a digestion, I wonder whether it could cause any kind of effect on the inflammation marker or something else. Thank you.
Thank you, Yan Yu. It was all 3 are excellent questions. First, always the question of egg and chicken. What comes first in terms of causing a particular disease? What is cause? What is the effect? To be honest, the answer is we don't have the answer as to what is causing inflammation and what is causing aging, because inflammation is not going to happen unless you have cellular damage. In this case, inflammation of aging, and cellular damage will not happen if you live long enough, because cells are continuously active. You can imagine, if you're a cardiomyocyte, you start beating when you are born and you're beating till you're dead. So those cells are producing these damage associated molecular patterns that eventually are sensed by sensed by the macrophages to cause inflammation. And that inflammation further impairs organ function, because it extracts a cost from an organ when those inflammatory cytokines are persistently increased. So I think they both go hand in hand, because we have shown that if you actually reduce NLRP3 inflammasome, you can delay the onset of aging. You can increase the health span. There is also data from the National Institute on Aging's intervention testing program that a drug that inhibits inflammation, NHDGA, actually increases lifespan. Okay. So that would suggest some cause, but I am more in the camp that they go, I think, side by side, because that cellular damage has to happen. I think your second question was, what fat is that? White fat or the brown fat? Yeah, that's all white fat. The visceral fat that we accumulate with age and with increased obesity is all white fat. Excuse me. And the visceral white fat is the one that is a major source of inflammation with aging, because of alterations in the immunological nature of the white adipose tissue. Your third question was, how can individual metabolism in people that is different contribute to effect or lack of effect of calorie restriction? That's a good question. We don't have the data, Yanyu, because this clinical trial was not designed to do that. So the way the people were selected was to exactly to address the question that you have, which is we don't want people to have different type of metabolism. We want everybody in the trial to be more or less the same, so that we can, understand what is really causal. So everybody that was in the trial was within the same age limit. They all were metabolically healthy. They were all normal. But indeed, that's, I think, a very important point because we know that some of us lose weight easily, some do not lose weight very easily. The energy expenditure between people can be different. Obviously, our gut microbiome, which contributes to changes in our metabolism, is different between different individuals. Therefore, I think what was really interesting from this clinical trial was how solid or how tight the data was. Despite our individual differences, individual genetic differences. Obviously, these are not inbred mice with 99% genetically identical, genomes. Still, these people, if you look at the transcriptomics, are very, very similar in their response to restriction of calories. I think additional studies would have to be done where you can, test people at the baseline to see how different they are, and then follow them with certain interventions, metabolic interventions, even calorie restriction, and how that affects the outcome of that particular intervention. I think those studies would potentially have to be done in future.
Yeah, that's great. And of time I lost, my last question is like, what kind of food you can suggest to people in general to promote the healthy aging? Because my study is trying to focus on the flavonoids or phytonutrients chemical, just to and not delay aging, extend the health living span for people. So do you, as a immunologist, do you have any recommendations for the diet or food? Thank you.
Yang Yu, I have zero recommendations because because I'm not a public health doctor who gives nutritional advice to people. And my job is to study and do science and study this intervention that extends lifespan. However, Yang, we should do what our grandmother told us, right?
Yeah.
if you eat enough vegetables, enough fruits, and if you eat a little bit less and walk a little bit more, we'll be fine. But I think humans find it very difficult to do these things. And I think we know the problem right now, at least in Europe and US, also in several developing countries, including India, is this new Western diet, which obviously has lots of negative effects because lots of saturated fats and fried foods. They
ultra processed foods.
Exactly, because our diets have changed in the modern world, and that is contributing to further increase in diseases. But I think the evidence, like you mentioned, flavonoids, I mean, these things are present in our vegetables and food and our fruits. And consumption of these things obviously helps. But what this trial basically is showing us is that if you activate the pathways that are going to act on your adipose tissue that stores the fuel,
yes,
if you activate the mitochondrial pathways that increase fatty acid oxidation, that is where I think the beneficial effects lie. That's where that's the reason why the benefits are there. And I think people have done that. If, for example, if people are in the same, if they consume same energy and they expend same energy, because in many studies, exercise has no effects, no beneficial effects.
Yes.
And the reason for this is simple, because unless you mobilize your adipose tissue by fatty acid oxidation, you don't really get beneficial effects. So it means that we have to be negative energy balance at least for some time.
Yeah, that's really informative. Thank you so much.
Thank you.
Thank you, Yang Yuan. I feel like he probably asked the question that a lot of people are wondering is sort of what can I do to limit calorie. It was interesting that you mentioned that it didn't seem like with calorie restriction, it mattered what kinds of food you were kind of
Right.
ordering,
Right.
and as long as you restrict. And I was wondering also, at least anecdotally, it seems like when people age, naturally your body tends to want to eat less, right? Your appetite decreases and um, things like that.
Right.
Is there some connection there? Is that a natural response of that's been developed over time in evolution that sort of maybe like the body is telling us like that is the way to go to increase health?
That's a very good question, Devin. I don't think I have the answer. I can only speculate. I don't think that's an evolutionary response. Because if you really look at human evolution, just till 2 centuries ago, the average human lifespan was what, 40 years, 45 years. So we just never lived long enough for those evolutionary mechanisms to engage, in my view. So, so I think what is happening, some people believe aging is programmed. I'm not very sure how programmed it is. I think lots of these things are stochastic because I don't think we've ever had enough time, because evolution is prioritizing reproduction. Our main purpose on Earth is to come in and reproduce and die. That's how the species have basically evolved. So, and living longer has not been the priority. So what happens? So if we know that elderly, one of the major problems, or at least the symptoms, is loss of appetite. People have anorexia. People don't eat enough. Whether that's beneficial or whether that's pathological, I am not very sure, in my view, because also if you look at the time when individuals lose their appetite, they've also lost substantial amount of muscle mass and overall body mass. So, an 85-year-old, 90-year-old person is not the same physiologically. So its fuel consumption, the requirement of energy it has is already less. But yes, I mean, in my view, it is a pathological response, because the things that people used to like, they no longer like. For example, whether that is part of the biology, it's not very clear, but this is certainly something that needs to be studied. I'm of the view that for example, when somebody is already restricting calories and is eating less with age, doing further of these things could potentially be harmful. So when these diet interventions are used, or how they affect people at different ages, is something that needs to be studied and is going to be studied soon.
Great, thank you. In the chat box, Henry had asked, there's a study conducted in Europe that says, human life expectancy can be up to 120 years. Are we expecting that? Are we expecting, discovering the fountain of youth and being able to live much longer?
I'm an optimist, Devin. I do think, I don't know about the fountain of youth. There are several people, I think, who are making lots of hyperbolic statements in our area, which I think is unfortunate, because we don't really have data to really say much at this point of time. The only thing we can say is that the modern science is making, I think, some significant advances, and it is very difficult to predict how science goes. I really don't know where the next big discovery is going to come from. Maybe some people have a special magic wand where they can shake it and find out what's the next big thing. I don't know, but I'm an optimist, and I think based on the work that is going on from several really exciting laboratories all over the world, I have a feeling that that overall that eventually we would have tools that would reduce our disease burden, and most likely, in my view, extend our ability to live longer. Whether that 120 years is the terminal limit is really not Sure, I'm not sure, because those are mathematical models. Unless you do a clinical trial, there's no way to know. And so you could say either way that, oh, you cannot live more than 120, or somebody can say, oh, you can live beyond 120. Fact is, nobody actually knows, and nobody will know for at least 100, for another 150 years till you do the study. And the people who are making those comments probably would be dead by then. So I'll end I'll end here with that remark.
Great. Thank you so much. I think because of time, I think we'll end there. Thank you so much for a fascinating discussion. And I would say quite a new paradigm for us to think about disease, not in terms of individual specific diseases, but just to think of aging overall as sort of that major risk factor. And I guess the main takeaway is just a small amount of calorie restriction can improve health, simply put. Thank you so much again, and thank you everybody for joining us. And I will unmute everybody so that they can all thank you personally.
Thank you, Devin. Thanks everyone, to all my colleagues. Many of you I know are watching this late in the evening. So thanks for joining. Thanks for your questions and your attention. And hopefully we'll have a chance to reconnect at some other time.
Thank you. Thank you, everybody.
Thank you.
Thank you.
Thank you for sharing.